Real quick lesson here on blood type genetics. So what we want to be able to do after this lesson is utilize a Punnett square to be able to predict offspring's blood type and percentage likelihood. Number two, we want to be able to answer questions of paternity based on blood type. All right, so how do we do it? Well, what we want to be able to show are the allele contributions of a mother and a father. So we could say the possible combinations of selected alleles on sperm and egg can be shown using a Punnett square, a diagram for predicting the results of a genetic cross between individuals of known genetic makeup. So the known genetic makeup we're going to use is the blood type of the mom and dad to come up with our answers. Now, if you remember from your first biology class, capital letters represented dominant alleles and lowercase letters represented recessive alleles. Now, in the case of blood type, what that means is that we're going to be able to use IA as a dominant A allele, IB as a dominant B allele, and lowercase I for the O allele. Additionally, all sperm alleles will be lined up on one side, while all egg alleles will be lined up on the other side. Every possible zygote genotype will be placed within the squares. Let's go ahead and work through an example of a Punnett square. First thing I'm going to do is I'm going to draw a large square, and I'm going to go ahead and break it into 4 quadrants. So you can put a line down the middle vertically and a line down the middle horizontally. We're going to arbitrarily pick sides for mom and dad. It doesn't matter. Let's say mom will take the top side here, and dad will take this side here, going down the left-hand side of these squares. Now, remember that blood type is ultimately the result of receiving 2 alleles from your parents. So we're going to go ahead and we're going to hypothetically cross 2 individuals using this Punnett square to determine what their probable offspring would be in terms of blood type. So let's imagine that mom has blood type AB, and let's say that dad has blood type O. Now, if you can recall, we're going to use that letter system to help us. We can do that first, and then we'll go back and look at it more on a simplified basis. But if mom has the genotype AB, that means that she has IA and she has IB as the 2 alleles that codes for her blood. Meanwhile, dad has blood type O, which means he has 2 lowercase i's. He received the recessive alleles from his parents, which code for blood type O. Now, when we use a Punnett square, we basically drop what we find on the top of the boxes down to each of the boxes below, and we push whatever on the side here into the boxes to the right. And we try to stick with the dominant trait if we know it first, followed by the recessive if it winds up being a heterozygous condition. But let's just go ahead and we'll start dropping these letters into the proper boxes below them or to the side. So here we go with our IAs and our IBs. And for dad's contribution to the next generation, we have all lowercase i's. Now, what does this mean as far as this box we filled up? Well, if we look at the potential offspring for mom and dad here, notice that 2 of them are identical. I, with a superscripted A, the allele for A blood, plus the allele for O blood, means that the dominant trait, the A allele, will match the recessive allele. So 2 out of the 4 are going to have a 50% chance of having A blood. Or we could say every offspring has a 50% chance of having A blood. Meanwhile, these 2 boxes here with the IB lowercase I means that there's a 50% chance that an offspring will receive B blood. Notice the absence of the other 2 blood types. There's no possibility for a child to have AB blood, and there's no possibility for a child to have O blood. So what we could say here is that 50% of the offspring would be A, and 50% would be B. Or to fine-tune that statement, we could say every offspring has a 50% chance of being A-blooded and a 50% chance of being B-blooded. Now, sometimes these letters tend to confuse students as they're trying to learn it. So we don't have to use IA and IB if we don't want to. We could basically use the double combination of letters for the actual genotype as well. For example, we know that mom is AB and we know that dad is O. So I could just put two O's there as well. And if that helps you stay a little bit more organized and it allows you to see the results better without having to translate them back, you could do it this way as well. So again, 50% A and 50% O. Let's look at another few examples. For our next example, I want to cross two parents. One who is A blood, and we know that parent is heterozygous. So what that means is that they have AO, or using the official symbols, we have I with a superscripted A and the lowercase I. And we're going to go ahead and cross that with a parent who has B blood. But it's unknown if this other parent is homozygous or heterozygous for B blood. So what we're going to have to do, if that's the case, is we're going to have to figure out both scenarios. So here we have I superscripted A, the allele for A blood, with the lowercase I allele. And we're going to cross that with one parent that's homozygous. Remember, that means you have two of the same allele. And then we'll also do a cross of that A-blooded heterozygote with a B-blooded parent who is heterozygous themselves. So what that will wind up looking like, we'll go ahead and we'll draw a couple of Punnett squares again to go ahead and do these crosses. And we can go ahead and put the one parent, again, it doesn't matter which side we put it on. We're going to separate their alleles per box. So this will be this parent right here, and this will be this parent here. We can do the same thing on this side over here. And over here, we'll take this parent, put that parent on the top. And we can see, is there a difference between the percentages of offspring and what they could have in terms of blood type between the two crosses based on whether one parent, the B parent, is homozygous or heterozygous? Let's experiment with our Punnett squares and see what happens. So we have I sub B there. We drop I sub B, lowercase I. And then if I go over to the second cross, I have IA, IB. Two lowercase i's for the next box. Okay, so let's go ahead and compare our results here. For the first cross, we have one possibility of our offspring having AB blood. We have another one, and that is a B. Sorry, it looks a little rough. There we go. We have a second one with AB blood too. So any offspring from this cross would have a 50% chance of having AB blood. Now down below here, the other possibility is just B blood, because remember the B allele is dominant over the lowercase I, which codes for O. So in this case here, with these two parents, we'd have a 50% chance of a child having AB blood and a 50% chance of a child having B blood. In this cross over here, notice we get some very different outcomes. We have one offspring could be AB blood, one could be A blood, one could be B blood, and one could be O. So for this cross here, we wind up with all four blood types being a possibility, with nothing more than one of the parents being homozygous for a blood type versus another parent being heterozygous for a blood type. So we have 25% AB. This box here would be 25% A, 25% chance of B blood as well, and a 25% chance of O blood. So these two parents right here, both of them being heterozygous, could lead to all four of the blood types. Meanwhile, this parent here, who was homozygous for the B allele, has a 50% chance of having an AB blooded child and a 50% chance of having a B blooded child. So if these parents here had a child that had O blood or A blood, we would then have to investigate further and ask the question, what happened where a child now believed to come from these two parents has either O or A blood? Was there a mix-up at the hospital? Or perhaps is the father not really the father in this case? Last example, let's go ahead and cross a homozygous B-blooded individual with an O-blooded individual. Now remember that homozygous means that you have the same allele twice. So if we have a B-blooded person who's homozygous, it's going to be IB as their code. And we're going to cross them with an O-blooded individual who is lowercase I. And we're going to take a look after we complete the Punnett square. What are the possibilities of offspring here? So we'll choose one side for the parent, IB, and then on the other side, we'll put the parent with the recessive alleles, lowercase I. So we'll go ahead and we'll drop the letters into the proper boxes to see what our offspring look like. So in this case here, notice that we have 1, 2, 3, 4 out of 4 boxes with the heterozygous B-blooded genotype. So that means that any children that these two individuals have will have, first of all, B blood, and they'll all be heterozygous for that B blood. So ultimately, they have B and O alleles, which means that the children here, if they were to reproduce at a later time who also was carrying one of those recessive alleles, it's possible they could have a O-blooded child later on somewhere down the line. So I hope these examples helped you understand how to create a Punnett square by using the alleles that each blood type carries. Put them in a box, see what turns out, and be able to predict the percentage chance of offspring to inherit a certain blood type. I hope you learned from this, and we'll see you next time.